So, what I think we'll start with today is the interactions between different immune cells. So, we have the interaction between macrophages and lymphocytes, also the effects of the cytotoxic and regulatory T cells, and then also how T and B cells are activated. Now, if you have, sorry, and after that, if we have more time, then we can also start with the antibody structure and function. So you don't really need to remember the like structure of an antibody like in depth. There's a couple of things you need to know, but not everything, and not everything in depth. And then maybe we can also discuss immunoglobulin subtypes. Maybe not memory, but yeah. So let's see how far we get. Okay. So the first interaction that I want to tell you guys about is the interaction between macrophages and lymphocytes. Now, Th1 cells. Okay, actually, before I talk about Th1 cells, do you guys know what Th1 cells are?
Helper T cells, right?
Yeah. So they're a part of helper T cells, and they're a very specific type of helper T cells, which help in the activation of macrophages and then also the cytotoxic T cells. And then what other name do we have for cytotoxic T cells?
CD8.
Yes, CD8 T cells. So all of these are different subtypes. So you have Th1, Th2, Th17, and T regulatory. So Th1 is the subtype which gives you macrophages and cytotoxic T cells, which are CD8 positive T cells. So what happens is these Th1 cells, they secrete a molecule called interferon gamma, and interferon gamma then allows the activation of macrophages and monocytes. Now, there is one important thing that you need to remember, and this is not specifically for the interaction between these two molecules, but it's more about macrophages. So, what's the difference between a monocyte and a macrophage?
So monocytes are being produced from the bone marrow, and then the monocytes become the macrophages in the tissue.
Yes. So, monocytes are present in the blood, and then if they leave the blood and they go anywhere else, they become macrophages. And then, even these macrophages, based on which organ they're in, they can be called a different name. So let's do a quick exercise. Where do you think will you find microglia?
In the brain.
In the brain, and they're macrophages. Now, what about Kupffer cells?
Liver.
Liver, dust cells? That's actually alveoli. In the alveoli, the macrophages are called dust cells.
Oh.
Yeah. Yep. So these Th1 molecules, not molecules, Th1 cells, they secrete interferon gamma, and this interferon gamma then activates the macrophages and monocytes. They're the same thing. Now, uh, sometimes even after a macrophage has been activated by interferon gamma, it can be an air quotes supercharged. So that means it will be much more active, and this can be done if the T cell binds CD40 on the macrophage. So what does CD40L and CD40 on macrophages mean? Basically, there is a molecule on the T cells which is called a CD40L, which is a ligand. Now, any kind of receptor, it has two components. It has the receptor itself and it has the ligand. Now, the ligand is the part which attaches to the receptor. In this case, the T cells, they have the ligand, which is this part, and then the macrophages, they have the receptor. So when there is a binding of the CD40 ligand and the CD40 receptor, the macrophages will be supercharged. They'll be more aggressive for phagocytosis. Okay. So, any questions about the macrophage and lymphocyte interaction?
No, we are good. Thank you.
Okay. And oh, actually, this part is also very important, and we will learn about this here. But the idea is that macrophages, when they phagocytose a random bacteria, they have the ability to present the antigens from that bacteria to lymphocytes. And when they present antigens to the lymphocytes, those lymphocytes can get activated themselves. So it's a constant loop. Let's say a macrophage phagocytoses a bacteria, it presents the antigen to the lymphocyte. Maybe then the lymphocyte attaches the CD40 ligand to the macrophage, supercharging the macrophage, or it secretes interferon gamma, which activates the macrophage to go phagocytose other bacteria. So it's a loop. Next, we have the cytotoxic T cells and then the regulatory T cells. Now, cytotoxic T cells, as the name says, they're responsible for cell death. Now, cyto means cell and toxic means toxin or harmful. So these cells can kill any kind of cell. And now, how do they do this? They do this by two mechanisms. One mechanism is they can induce apoptosis, and then the second mechanism is that they can release granzymes and perforins. Oh, yeah, it's here. So, perforins and granzymes, these are the two mechanisms. Now, do you guys remember another cell which can use the same method of killing using perforin and granzymes?
Sorry, Dr. Taha, I didn't get it. Can you ask me?
So there is another cell, another immune cell, which can induce cell death by perforins and granzymes. So do you know what
The
cell
natural
that
killer
is?
cells.
Yes, natural killer cells. So they can also use perforin and granzymes. Actually, they can also induce apoptosis in cells. So they work pretty similar to cytotoxic T cells. It's just that the way they detect uh, infected cells, that is different. Now, this is very important to remember that cytotoxic T cells, which are CD8 positive, always bind to MHC1. And when we look at the activation criteria, we'll understand like what is the importance of MHC class 1 and MHC class 2. Next, we have these regulatory T cells. Now, as the name says, regulatory, that means they regulate the entire immune system. They can do multiple different things. They can activate the CD8 cells, they can activate macrophages, they can activate the NK cells. See, all of these are different regulatory cells. Oh, sorry. Yeah, all of these are the helper cells, and specifically, these are the regulatory cells. Yeah. Okay. And, I would say one thing that you need to remember about these regulatory T cells would be remember these two cytokines that they produce, interleukin 10 and then TGF beta, tumor growth factor beta. These are actually anti-inflammatory. A majority of the cytokines that the body produces are inflammatory in nature, but these are two which are non-inflammatory. So these are the ones you need to remember. Additionally, this molecule, the FOXP3, just remember that FOXP3 is involved in a condition called IPEX. Now, I mean IPEX, it basically means immune dysregulation. That means you have some kind of autoimmune problem. You also have polyendocrinopathy. That means multiple different endocrine organs are being affected. It's also affecting the GI system. That's why it says enteropathy. And then it is X linked. And That is what IPEX syndrome is. So it's an autoimmune condition which can affect endocrine systems and GI system, and it is an X-linked condition. So that is all you need to know about IPEX. Majorly, I'll tell you to remember the molecule involved, FOXP3, and then the name of the condition. This is a rather rare condition in the USMLE, so you won't really encounter it, but it's good to remember the name and the molecule or the, yeah, receptor associated with it. Okay. Next, we have the activation of the um, T and B cells, or the T and B lymphocytes. Now, we don't really need to look at this wall of text. Instead, we can simply focus on the image. So, let's say there is an antigen, and that antigen is present on a bacteria. Initially, what will happen is an antigen presenting cell, any kind of cell which can present an antigen, will directly phagocytose the bacteria. It will break down the bacteria, and after breaking down the bacteria, it will post it onto the MHC class 1 or the MHC class 2. Now, based on which MHC class this molecule is posted on, will depend like what type of T cell it is it. So if it's a MHC class 2, what kind of T cell do you think would be involved?
TH1,
Yeah,
CD4.
CD4 positive cells. And then if it's an MHC class 1, then which T cells would be activated?
CD8.
CD8. Yes, perfect. Now, both of these cells, right, CD8 and CD4 cells, they have a molecule which is called a T cell receptor. This T cell receptor would be the one which directly connects to the MHC molecule, and that allows for sharing of information from one cell to another. Now, how did this happen? The cell initially took the antigen, broke it down, gave it on the MHC complex, and then the T cell receptor took in that uh, molecule or the protein which was presented on the MHC complex, and then the T cell got activated. Now, there is one more condition that you need to remember. That is this part. There is a connection between B7, and B7 is the short like acronym for CD80 or CD60, and that binds to CD28. Sadly, this is something you will need to remember, but this is a secondary action, and once both of these actions are met, only then can a T cell be activated. So any questions about the activation of T cells?
No.
No? Okay. So then, okay, a couple of questions. Uh, how is, not how, but what kind of molecules are presented on MHC class 2? Endogenous or exogenous?
For class 2 would be exogenous.
Yeah, for class 2, it's exogenous. And then for class 1, it's endogenous. Now, let's take this example here with the antigen which was phagocytosed and then presented. Or rather, not phagocytosed, but let's say, okay, yeah, let's use the same thing. A bacteria was phagocytosed, and now the antigen is presented on the MHC complex. So, which complex would it be presented on? 1 or 2?
If the antigen is coming from outside, you said?
Yeah, if the antigen is coming from outside, then which MHC would it be posted on?
1.
1?
No, sorry, 2.
Yeah, so it will be posted on 2. And then what example would you be able to give me if I were to tell you, give me an example of endogenous proteins, which would be
That would
presented?
be for endogenous, that should be viral viruses, right?
Perfect. And why do viruses have endogenous proteins?
So they are the one hijacking the system inside of a cell, and
Yeah,
I think something like that.
that's true. So what a virus does is it integrates its DNA or RNA into the host DNA or RNA, and then it starts So not starts, but it hijacks the body and it tells the body that instead of producing your own DNA, I want you to start producing my DNA and my proteins. So then the body complies. It starts producing the viral proteins and DNA, and then the cell, it gets so swollen up with all of these extra proteins that it bursts open. And when it bursts open, it releases a lot of viruses into the blood or into the surrounding tissue. And all of these small viruses continue the same thing.
Okay.
Now, okay, so we said that the APC ingests and processes antigens, and then it migrates to the draining lymph node. Okay, because think of lymph nodes as those checkpoints. So any kind of infection that the body identifies, it will immediately go to the closest lymph node because the lymph node connects the entire immune system. So if you want a full blown immune response, then all of the immune cells should have the information about what the antigen is that the immune system is fighting against. And to do that, any cell which encounters a foreign antigen will go to the lymph node and it will follow the lymph drainage and it will give its information to as many lymph nodes as possible. Now, for activation signal 1, which is an exogenous antigen, it's presented on MHC2. It's recognized by CD4. So that is what we've been talking about so far. And then if it's an endogenous protein, then it's presented on MHC1, which then activates CD8 cells. Now, for the proliferation and survival, which is the secondary activation, that is with CD80 or CD86 and CD28. And then finally, when the cell has been activated, then it can start doing what it does. So either it can produce cytokines, maybe it activates other cells, maybe it starts cell induced killing, which is the cytotoxic T cells. So basically that T cell would be activated and now it has the ability to carry on with its functions. Next we have the B cells. Now the B cells, they're usually only activated by a CD4 positive cell. Now what the CD4 positive cell does, it actually happens in reverse. Here, let's say this is a CD positive cell. There was some molecule which an antigen presenting cell presented to the T cell, and now the T cell has the molecule from the MHC complex to the T cell receptor. Now that cell will take the same molecule and go to another B cell which has a MHC class receptor. Okay.
Sorry, Dr. Taha, I was distracted. What did you ask?
Okay. No, no. I was just saying, like, if you understood. So basically, what happens is you have an activated T cell. Yeah.
Okay.
And now, when we talked about B cells, we talked about like not B cells, but when we talked about the recombination, we talked about like how your body can produce almost any kind of antibody, and it has the antigen library for all the possible combinations. So
Okay.
this activated T cell will try to find a particular B cell which has the exact same antigen presented on its MHC2 complex, and it will try to find that complex.
Okay.
And then once that has been done, it will give a secondary response. In this case, it's the CD40L and CD40, which is the CD40 ligand and then the CD40 receptor. That is the secondary response. And then, it might release some cytokines, and that leads to the activation of B cells. So here, if we quickly read, it's that the TH cells, they're activated regularly. After that, the B cell receptor mediates endocytosis, and then the exogenous antigen is presented on MHC class 2. Okay, so that's pretty much what we've discussed. Now, the MHC class 2, it's recognized by the T cell receptor on the Th cell. After this has been done, the interaction is CD40 and CD40 ligand. And then finally, there is one thing that happens is that the Th cells or the CD4 cells, they secrete some cytokines that determine the immunoglobulin class switching of B cells. So it basically just tells the B cells, these cytokines, they basically tell the B cells which type of immunoglobulin to release. Is it going to be a IgM? Is it going to be a IgG? Or is it going to be an IgA? And then finally, when everything has been done, they start producing the antibody. Now, what kind of antibody do you think is IgM? Is it an acute antibody or is it more of a chronic antibody?
It's an acute.
Yes.
M for acute, G for chronic.
Yes, M for acute and G for chronic. And then, okay, tell me if this statement is correct that all hepatitis can have chronic infections.
No.
No,
Incorrect.
not Hep A.
Yeah, that's correct. Not Hep C, because Hep C does not have a chronic infection.
No, A. Hep
Yes,
A doesn't have.
yes, Hep A does not have a chronic infection. Actually, Hep C is the most common to have a chronic presentation.
Yeah. You're trying to trick us, huh, Dr. Taha?
Yes.
Yeah.
So you guys understood how these cells are activated?
Yeah.
Perfect.
Yes.
Okay. Next, we have the structure of the antibody, and there is okay. I'll tell you there is out of this entire segment, there is like one or two things you need to know, and they're pretty simple. Oh yeah, you also need to know about these, but okay, those are different. That's not the structure of the antibody. Okay, there are two things you need to remember. Remember that if this is an antibody, this is the fixed component of the antibody. This part here, it's the fixed component because for any type of antibody, this will remain constant. Any antibody you pick up will always have this structure. Actually, it will also have the hinge. All of that will be constant in every antibody. Now, this portion here, that is where these antibodies will differ. This portion would be different in almost all antibodies. That is the exact like binding region, and it's called the Fab region. Fab region can be variable, but then the FC region, that's the fixed component of the antibody. That is what you need to remember. Now you also need to remember that sorry, you need to remember that wait, the market here. Oh yeah, yeah. So you also need to remember that the top part, so this entire part is the heavy chain, and then the parts which are at the end or like towards the side, they're called the light chains. So if you know that, then you really don't need to remember all of these other things. So any questions about the structure of the antibody?
No. But a quick question, Dr.
Yeah.
Taha, on affinity and avidity. There was a question on UOL, so they were asking about the affinity and avidity. So what I understood for avidity, meaning there's more sites, binding sites for the antigen, and the affinity is the strength, the joining strength for the antigen and the antibody. So I was kind of confused on that. Can you please elaborate on that a little bit?
Yeah. So affinity, it's as you said, how strongly does a antibody attach to the antigen? How strong? And then avidity is how many of those regions can the antibody bind. So let's say here the antibody can only bind two regions or two antigens. But let's take an example of a IgM. Now, see how many sites it can bind. So many. So IgM will have a much higher avidity compared to, let's say, an IgA, which only has two, or an IgG, which only
Okay.
has, oh sorry, IgA has four, but IgG, which only has two. So
So that would be
yeah.
highest avidity on IgM because it has more binding sites.
Yes,
And for the affinity part?
and affinity, I would say IgG would have the most affinity, and then IgM would have the least affinity. Yeah,
Okay. Thank you.
no problem. Yeah, and now these are the different things that antibodies can do. One, they can neutralize a bacteria. Now, neutralization, it basically means that the antibodies have just covered the entire bacteria, and now the bacteria can't actually do anything. So now, even if it wants to stick to the epithelium, it cannot. If it wants to invade the epithelium, it cannot because it's surrounded by these antibodies. And eventually, because the cell cannot do anything, it will die off. Next is opsonization. Now, opsonization, as I told you, is when one antibody, it can bind multiple different bacterias, and then when it binds multiple different bacterias, it will allow for it will allow for easy phagocytosis. Now, here the example that they've given is a little different, and here the example is that all of these antibodies, they're actually on the cell surface, and because they're on the cell surface, they just completely enclose the bacteria, which allows for easy opsonization. So those are the two variations of opsonization. And then finally, the third variant that we have, or the third function of antibodies, is that they can activate complement. Now, complement, what do you think? Is it adaptive or is it innate?
So the complement would be adoptive, right?
No, so complement is actually part of the innate immunity, and there's only two cells which are present in the adaptive immunity. It's the T cells and the B cells. So
Okay.
only those are present in uh, adaptive immunity. Everything else is in innate immunity. And
All right.
then finally, the complement, it has a lot of functions. It can do everything. It can do opsonization. It can do cytokine production. It can directly kill the cell. It can cause endocytosis, not endocytosis, but it can cause chemotaxis so that it recruits more cells. So it can do all of these things, and we will learn about them, yeah, in complement. So there's three pathways of activation, and then yeah, this is the pathway for complement. You don't need to remember all of this, so that's good. There's only like one or two things that you need to remember, and some proteins. But those are the three things that antibodies can do. They can neutralize a bacteria, they can opsonize a bacteria, and they can activate complement. Now, I have one more question. Uh, Antibodies, they have a lot of variations. So, the same IgG antibody, which can fight Staph aureus, can also help fight, let's say, hepatitis B. Not the exact same, but the same class. So, why do you think the same class of immunoglobulins can attach to both Staph aureus and hepatitis B? Not why, but rather how.
So, sorry, I didn't get it, Dr. So which, how they're attaching hepatitis
Yeah,
B, you
I
said?
just want to, so I can simplify the question in a way where why is there so much variation in these antibodies? That IgG, it can, okay, I can draw this for you. Let's say one IgG has a binding site which looks like a square. One IgG has a binding site which looks like a triangle. one has a binding site which looks like A star. So why or how is there so much variation?
Genetic recombination?
Yes, but there is one very specific keyword that you need to remember.
Somatic hypermutation or
No,
hyper
it's
something.
three letters.
Not that.
It's three letters.
VDG?
VDG?
Yes, V, D, and J.
VDJ?
Yeah. The VDJ recombination is actually what allows these antibodies to have so much variation. Now, it's similar, like what your body does when it's developing during conception is your body produces all the different types of antibodies. So, if we were talking about shapes, let's say it produces any possible shape which is like conceivable, and it stores all of these. So, whenever you need any kind of antibody, it's already been produced. You just need to find and activate that cell. So that's the
So
yeah.
then Dr. Taha, what is somatic hypermutation? Isn't it the same name for VDJ? Genes been
Oh
made?
yeah, that it is a relatively. Oh yeah, it is actually the same name. Somatic hypermutation is also called the VDJ. Yeah, that's true. Both
Okay.
of them are pretty much the same. Oh yeah, they say somatic hypermutation. But I would say with somatic hypermutation, it's more like VDJ is a part of somatic hypermutation, but somatic hypermutation isn't a part of VDJ. Where like somatic hypermutation is a bigger umbrella, and under that you have the VDJ classification.
So VDJ is part of somatic hypermutation.
Yes, it's a type of somatic hypermutation, you could say.
Okay. All right. Thank you.
No problem. Now we have immunoglobulin subtypes. So obviously there's G, A, M, E, D. Now, a couple of things here. One thing that you need to remember is that these two immunoglobulins, which is M and D, they're expressed on, sorry, yeah, this part here. M and D immunoglobulins, they're usually present on immature B cells. And as they mature, they lose these. Oh yeah. So affinity is the antigen antibody, while avidity is the cumulative binding strength of all antigen antibody interactions in a pentavalent molecule. So as we said, so just to recap, there is a lot of different immunoglobulins. You don't really need to remember about D, but then remember that IgM and IgD, they're present majorly on these mature and naive B cells, or mature and immature B cells. And then yeah, here this is pretty basic things. IgG, that's responsible for chronic inflammation, and it's the most abundant immunoglobulin subtype which is present in serum. IgA, it's usually present in mucosal surfaces, and if they mention about colostrum, no, but also breast milk has a very high concentration of IgA, so that is important to know. IgM, that's a acute phase antibody which is produced, and it has the ability to bind multiple different antigens, so it has the highest avidity. uh, IgD, it's not normally present in the blood, it's only present on the surface of the B cells. And finally IgE, it is responsible for allergy mediated like reactions, so asthma, anaphylaxis, and all of those, and these are released by eosinophils. So, any questions about these antibodies? I know I went through them very quickly, but there's not a lot that you can extrapolate from this information, which isn't very basic knowledge. Hello.
Yeah, sorry.
Yeah.
Yeah, it's following. Thank
Oh,
you.
okay.
Yeah.
So so next we have antigen types and memory. So wait, does it have types? No. Okay. So basically, there's two different types of antigens. One is thymus dependent, and the other one is thymus independent. Now, the difference between these is that if any kind of antigen has a protein, or it has a specific molecule, usually like a protein, that will give you memory cells. And then if you do not have a protein attached to a certain molecule, let's say it's a normal bacterial infection with an LPS, so it doesn't really have a yeah, it doesn't have a protein attached to the LPS, so your body cannot remember it, and that's why it's thymus independent. That means it doesn't go to the thymus. Now, which one of these do you think is a part of adaptive immunity? Is it the thymus dependent or the thymus independent, which is a part of adaptive immunity?
So for you were saying for the memory cells, right?
Yeah, so thymus dependent is memory cells. Thymus independent is all the general infections that we have. So
Okay.
then which one of these is adaptive immunity?
Memory cell?
Dependent.
Yeah. So memory cells and dependent, that is the part of the adaptive immunity. So now let's say I want to give someone a vaccination. So what type of so not what type, but which of these should I prefer? Do I prefer the independent or the dependent if I want to give someone a vaccination?
Dependent.
Yes.
Dependent.
And why do you think dependent?
So if you're introducing the vaccine with the same antigen, our body has being familiarized with it, right? So it's not gonna attack.
Wait, it's not going to attack?
I mean, attack and kill the vaccine.
Oh yeah, so you're so basically, when you give someone a vaccine, you can think of it as it's like a weaker version of the virus, and the body fights it and remembers it. And then when the stronger version of the virus comes in, the body remembers that, oh yeah, I fought the weaker version, and I know exactly what to do to fight the stronger version. So it can immediately mount a different attack. And I have a good illustration of this, and I will send it in the group.
Okay.
Yeah. Okay, so then I say we will continue with the complement and maybe complement disorders and okay, cytokines you have to memorize. Okay, so hopefully by tomorrow we can complete everything till vaccinations. Then we might spend one day for vaccinations and uh, reactions. Okay,
Okay.
okay. So let's end here for today, and just give me 1 minute.